My name is Arun Konagurthu. I'm a senior lecturer in the Monash Faculty of IT. My main research area can be classified as structural bioinformatics. I deal with problems that arise in biological data, mainly macromolecular structures, proteins, DNA, RNA. But I am a trained mathematician and a computer scientist. My undergrad was in mathematics. My PhD was in computer science. And I was interested always to work in an area that's at an intersection of computing and biology in some sense. Advances in techniques to resolve three-dimensional structures of proteins have arisen in the last half of 20th century. And there's a whole growing mass of protein structures that have been elucidated, whose structures have been resolved, and we can see them to atomic precision. And to understand the general principles of protein structure and architecture is something of crucial interest to drive major advances in biology and medicine. Computing is central to addressing these problems. And I am interested in applying aspects of mathematics and statistics and discrete and continuous optimization problems that arise from computer science literature to address these problems. So that's pretty much my main approach to problems that arise in biological data.
So your general research area would be computational biology, a specific aspect of that. What is computational biology?
Well, I guess it's a loose denominator for a whole amalgam of problems that arise in biology. Biology was initially fairly anecdotal and experimental, but I guess with the growth of computing, with the powerful computers that we have today, it has become quantitative and precise. Well, one aspect of computational biology deals with these macromolecular data, but there's also other aspects of computational biology, for instance, trying to understand, simulate, and model the spread of diseases, epidemiology, trying to understand the interactions between predator and prey, ecological modeling. The whole gamut of things can be classified as computational biology, in my opinion.
Okay, so tell us about stringology and how it relates to understanding protein structure.
Stringology is a very curious term to describe quite a serious research area which involves theory of processing strings. There are a lot of problems that arise in data that are fundamentally can be described as sequence of categorical data which have some strict precedence associated with it, linear ordering constraints. A classic example would be the blueprint of life genome is a long sequence of text, can be represented as a long sequence of text, which is roughly 3.2 billion letters for a human genome, representing the 4 nucleic acids that make up DNA. And given the mass of data that exists, computer science is interested in trying to find certain patterns in this data. Stringology arose out of trying to consolidate the theory of trying to do, let's say, for example, pattern matching on large texts efficiently. Well, I guess the correct term should have been string theory, but that's propriety of physicists. Unfortunate thing about stringology is it sounds like a cult, something that Hollywood stars subscribe to. But I assure you there's a lot of serious computer science behind stringology. And there's also a beautiful conference called Prague Stringology Conference. Prague is a beautiful city to go to if you ever want to research in that area and publish papers there.
So if you did sign up some Hollywood stars, you'd get better funding, wouldn't you?
But it won't be science, it'd be Scientology.
So tell us why you prefer to use Bayesian-oriented statistical methods for your work.
So I guess Bayesian view, I suppose in what I understand, interprets probability as a measure of state of knowledge or degree of belief. And it can that statistics, that form of statistics, can be thought of as a natural extension to rules of logic. It can be very useful to as an extension of rules to logic when reasoning under uncertainty. Okay, and I think given the fact that it's got some very good axiomatic basis and an intuitive way in which probability can be defined, I suppose it has a lot of traction in a large class of scientific problems which can be described fairly rigorously based on the observable evidence that we have. And hence, I think one of the prime reasons for its popularity and one of the reasons why I use it is effectively trying to make inferences on observed data, and it's a very reliable method of doing it. I don't know of anything else that does it.
Actually, it would be useful to me if you would take just a couple of minutes to explain two-part message lengths and how that relates to Bayesian inference.
The two-part message length can be effectively rationalized as a product of Bayes' theorem and the Shannon's information content, the notion of what the rule of what is described as the measure of Shannon's information content. So if you take the product rule, I mean, even if you, Bayes' theorem is an expression of product rule. If you take the product rule, if you have an hypothesis H and a data D, the joint probability of the hypothesis H and the data is the prior on the hypothesis times its likelihood. It's same as saying it's the prior on the data times the posterior. And often, in most of the inference problem, model selection problems, you want to find a hypothesis that maximizes the posterior. But if you are completely independently, or seemingly independently, Shannon proposed that if there is an event E whose probability is P of E, then the shortest code to represent that event would be the negative logarithm of that probability. Now, if you applied Shannon's insight to the product rule, you would effectively get the Bayes' theorem restated in information theoretic terms. So the information, or the joint information in the hypothesis and the data is equivalent to information in the hypothesis plus the information in the data given the hypothesis. And you can rationalize this as a communication process between an imaginary transmitter and receiver pair, where in the first part, I tell you the information, I must say that the transmitter, me, has some observed data. And I have to send the data to you, to the receiver in a lossless way. And I send a certain hypothesis. It could be a model over a continuous parameter space. If it's, I'll pause there. How much detail do you want?
Models are fine.
Okay, so if you have a model, I would send the parameters of that model, and the statement of that would be the first part of my message, and that takes a certain number of bits to state if it's sent over a classical channel like the internet. Or, and the second part would be the data, the transmission of the data given the model. And that would be the second part of the message that takes a certain number of bits. A two-part message is the total lossless transmission required to state your opinion and the data given the opinion.
So how does that deal with overfitting?
So it is a model with more free parameters can describe data much better than a model with fewer free parameters. So it's effectively a trade-off between the first part of the message, which deals with the complexity of the hypothesis, and the second part of the data, first part of the message that deals with the complexity of the hypothesis, and the second part of the message that deals with the data given the hypothesis. So what the minimum message length principle tries to do is effectively find the model whose two-part message length is minimum. So there's a trade-off, there's implicit trade-off between how complex the model can be and at the same time explain the data with some with fidelity. So, and it is in that two-part description that the trade-off can be addressed quite naturally.
So this theoretical principle is something you can actually apply in practice in searching for protein structures or string matching or what have you?
So there's a general part to that question and a specific one to how it's applied. The general part to the question is, it's been fairly explored, MML principle, in the strict form, can be solvable on something like a binomial distribution in quadratic time. If you have, when I talk about a binomial distribution, it's effectively, let's say you have a set of n coin tosses and you want to infer the probability of the head of that trial, then you effectively can have a strict way, absolutely watertight way of inferring the probability of head under the two-part message framework. However, the tractability of this strict formulation becomes unviable when you go to even multinomial distributions or or even in continuous spaces where your random variable is continuous rather than discrete. And then, however, there are approximations, quadratic approximations of MML. Especially one thing that comes to my mind is the Wallace-Freeman approximation, which makes under certain assumptions using a Taylor series expansion and truncation after the quadratic term, is able to effectively address the estimation under the MML framework, under the minimum message framework of the parameters to reasonable validity. I mean, they're very useful in real world settings. How do I use it in my work? In fact, more recently, my collaborators and I are interested in trying to infer mixtures of directional distributions. For instance, one directional distribution on a three-dimensional sphere is called a Kent distribution. We want to infer mixture of directional distributions on the entire known conformational space of protein coordinates. And I have a PhD student, a very talented one, who actually addressed this problem recently, and this work is under communication. And what a mixture model does is, in an unsupervised way, tell you how describe the entire conformational the distribution of all protein coordinates on a sphere using certain component Kent distributions, each with a certain probability, and each of that Kent distribution has its own 5 parameters. And all this has to be inferred in an unsupervised way. There's no supervised step in this process. And MML is quite useful. And the approximation that we've used is the Wallace-Freeman approximation to address that problem.
So tell us about the general impact of simulation on computational biology, the general direction of the field.
About systems biology.
Wherever Arun wants to take it.
I think Markov chain Monte Carlo models have had an impact on all of science, let alone biology. And it's through the use of MCMC techniques in biology that you can see how useful it has been, especially some of the fields that come to my mind. I mean, for instance, trying to MCMC techniques have, even with the search, MML as a search problem that I'm used to, poses a criterion. It doesn't tell you how to go about finding it. And it is through MCMC sampling that one tends to infer the parameters for the model of interest. Just going outside my own immediate scope of research, molecular dynamics is a very fascinating area of computational biology where, well, there's a problem called the protein folding problem, and if anybody who solves it can book his flight tickets to Sweden. People have been trying to address this problem since the birth of computing, essentially. And molecular dynamics arises trying to understand how a string, a chain of polypeptides, amino acids, fold into a native three-dimensional shape. And there's a fascinating amount of simulation that arises in that context. Most of them are done these days using some molecular potential energy functions, which is an amalgamation, which is sort of a mixture of things like Hooke's law, Coulomb's law, a few quantum mechanical equations thrown in, a potential energy function which is given to you, and you are sampling from some, let's say, Boltzmann distribution to effectively find a state which has minimum free energy. That's the classic Metropolis Hastings algorithm. So yeah, I mean, it is things that cross my mind are the influence of simulation would be in statistical applications to structural biology through the process of trying to infer certain conformational preferences. So if you want to understand if proteins prefer certain conformations over the others, you effectively use the power of random numbers, MCMC techniques, to kind of sample that space.
So what would success in protein folding even mean?
Effectively,
the hit rate on prediction, as I understand, is something, the last time I looked was just, I don't pay attention, 72% or something. So wouldn't 75% be a success?
Well, in fact, if you had a hit rate of 50%, you could sort of get, I mean, I think there's several layers to structure prediction, which is so protein folding problem is trying to understand how proteins fold from first principles of physics and chemistry. And that's tremendously useful because structural biologists spend half a decade trying to crystallize these proteins and shoot X-rays into them and try and infer their three-dimensional structures experimentally. So if you could have a computational process that actually is able to simulate this folding process, all the big pharmaceutical companies would abandon their wet labs and jump onto and hire data scientists to instead of structural biologists and chemists, I suppose.
X-ray crystallographers would be out of a job?
Well, they might just be, because there's an advance in imaging. So there's electron microscopy, cryo-EM as it's called, which is able to now image or sort of, we can resolve structure to low resolution, say about 10 angstroms. So you can resolve large complexes of proteins in assembly, like a ribosome. Ribosome is an assembly of several protein subunits. Might have about 1 million atoms in them. Just that molecule would have about 1 million atoms. You can actually the technologies are getting to a stage where you can image complexes at that level using the cryo-EM technique. So they might just be out of job not because of a computational person solving the problem, but because the technology has improved. But I suppose back to the structure prediction problem, the structure prediction has many layers to it. If you had to do it from scratch, it's called ab initio structure prediction. Success rates are nowhere near even 30%. And there is a worldwide competition called CASP, where structural biologists solve a structure, withhold the structure from being deposited, submit it to this competition, give just a sequence, and you have a whole mass of computational biologists trying to predict the structure. And there are assessors who would say, you've come close to it, or you've failed, and so on. And there's a whole biannual contest, which is very popular. But there's other forms of structure prediction which is knowledge-based, homology-based, where if you just give me the sequence of hemoglobin, let's say, and if the structure of hemoglobin were not known, it could well be that there is an homologous a protein which has descended from a common ancestor whose structure has been resolved. So I can borrow the information of that structure and start to meddle with it slightly to predict the structure of the target protein whose structure has not yet been categorized. So, and there the success rates are fairly high, even up to 90%. So there's a whole spectrum of how much knowledge you can put in and hence get more reliable prediction. But the ab initio version is not even 30%.
Okay, so what's the cutoff for the Nobel Prize?
I don't know. I'm happy with an Ig Nobel Prize.
I'd be happy with one too.